Lake Maxinkuckee is home to about 2,000 people and is vital to the economy of this town. It went through a crisis several decades ago in the form of eutrophication, which is a process that kills all creature in the lake due to an excess of nutrients and sediments. In order to save this lake from eutrophication, the Wilson Wetlands is constructed, along with two existing wetlands. Built by the Culver academies, the Lake Maxinkuckee Environmental Council, and DNR in 1987 as a part of the state conservation program, the large sediment trap, various islands, and long ditch to the lake are to help the water slow down and its nutrients and sediments settle, mitigating the process of eutrophication. Now it is 2019, more than 30 years since the construction of the wetland, and we wonder, is the Wilson Wetland still working as intended? In order to understand the importance of the wetlands, we need to first understand watersheds. They are an area of land in which all drained water goes through a common outlet, in our case being the three ditches, including the Wilson Ditch. These are the primary contributors to the water in the lake, and the quality of water in those ditches are very important to the quality of water in the lake, which is vital to our economy and health. Since most of the land around these ditches are farmlands, a lot of nutrients in the fertilizers would flow into the ditch, which is why wetlands are needed to control the flow of nutrients into the lake. In order to test our hypothesis, we conducted tests at 4 sites: the wetland inlet, which is where the water flows into the Wilson Wetland to settle; the outlet, where water flows into the lake inlet, then the lake, and eventually the lake outlet. The sediment and nutrients in the drain water should settle along the way, resulting in a healthy lake with no dangers of eutrophication. The wetlands inlet is where the water flows into the wetlands to be settled. The water quality should be the worst here.
It meant a lot to that spider.
I don't know what that was. What?
That spider's being
I'm literally scared of every creature.
I killed the weakest of its species.
Okay, great.
Keep going.
Shake it. Give me that nitrate. I already have it.
Hold on.
Thank you. Okay, Roger, do you want to do the nitrate?
And
And I time you? I was like, I don't know what you're going to scoop besides mud. Go.
Yeah, you might be able to collect any macros that are like
Swimming.
swimming.
Yeah. Shake it. You can. You wait, Dr. D.
Got it in my boot.
I just got a bag in my face. Ew! Oh my gosh, you guys are literally children. Me? No. Oh, I look like these two.
Yum, eat it.
Yum.
$5.
Do it for the vine.
$5.
I ain't gonna do it. Ah!
The wetlands outlet is where water with some nutrients settled flows out and towards the lake inlet. This should be where the water is the second to worst.
Alright, so we're walking to the wetland outlet to test the levels of everything and how clean the water is. How fast it is. Okay, you're ready? Look.
You gotta take a video of this.
So the water is flowing really fast through there, and it's not supposed to be doing that. It's supposed to be flowing like slowly, so it has time to like clean through everything. But that's bad. So it's flowing really fast because the wetlands like overflowed, and there's been a lot of rain recently, and so it's just flowing. This is all excess stuff, so the water in the lake will be very dirty. The pH level. Oh, this needs to be, dude. The pH level. Hold on, let me do my sheet. The pH level. What would you say that is? Did you say that's a 7 or an 8?
Definite 8.
8?
Alright, Raj Daddy. What exactly is the point of the pH test and what can it tell us?
So pH basically measures acidity in water, and a low pH means that the water is acidic. And when water is acidic, they are able to dissolve heavy metals such as copper and aluminum, which makes the water toxic, especially to aquatic organisms as they reproduce. So the pH level By measuring that, we can make sure we know whether the water is toxic or not.
So it's staying on the rock? Well, it's not wet, so
It's got moss
Do you know that water is wet?
water is water.
And it's wet.
No, it's not. It's between 40 and 100. So, but
Probably
like
100.
it's not.
Looks pretty bad. I can't
Hey
barely see
Topher, you
this.
want to give us an opinion? Is that Topher? Yeah, you're Topher, right? Yeah, you want to give us your. Well, I couldn't see like from the angle.
It looks like 100 to me.
Do you think so?
It's about the same, like, right.
You think so?
A bit worse.
Okay.
Like 80?
Oh, oops.
Only horizontal? God dang it.
Whoops.
So what exactly is the point of macroorganisms?
So, chemical tests are flawed in that they only provide a snapshot. They are data that are about the day that we did the testing on, so we are unsure if there are any external factors such as temperature, just humidity and all that, and how that's affecting the water quality of that day. But the macroorganisms test is a test that shows us how the water quality has been over time, so we can be more certain that it's not because of the specific day we did the testing.
Alright, good.
Get a little bit more. Literally just submerge the bucket. Here we go, we're about to reveal the big secret. Nitrate is 5. 5. Look at my hair. Okay, 5. See, 5, right?
Yeah.
Okay, cool, 5.
A flatworm?
Would you say 1 or below 1?
Like,
Okay, 1. Phosphate's 1. Cool. No, like it's not like working.
The lake inlet should be where the water flows into the lake, and since a lot of the nutrients have settled during its long journey from the ditch, it should have been the third worst.
Dr. Chen, please just express your wisdom and tell us all about what is the exact point of the dissolved oxygen test.
So, dissolved oxygen is basically the amount of oxygen that is within water, and dissolved oxygen is very essential for fish and other animals to live, and they are often an indicator of algal blooms because as dead algae are being decomposed, the bacteria that do that requires oxygen. So if the dissolved oxygen level is very low, it means that there has been an algal bloom and there is eutrophication in progress. And this is why we do the test to make sure that we know if eutrophication is happening or not.
The BOD stuff. We're gonna put it underwater, then take it out underwater, then take it out underwater, then put the cap on.
Yes.
Look at that, Raj.
So, Dr. Chen, as we move forward, what is the reasons for and why exactly do we do the BOD5 testing, and what does it tell us?
So the BOD5 is pretty similar to dissolved oxygen, as in that both are testing for that. However, BOD5 runs in the span of 5 days, so it gives us a clearer picture of the biological oxygen demand of the bacteria as they start composing dead algae.
Step. How big is it? That way. Okay, well, hold on. Let's make it bigger. Hold on a second. Let's make it bigger one this way. Okay. And then we're just gonna. Okay, well, hold on a second. It said after this. We're just making sure to hold this. That's a little above 5. So add 1 nitrate wide thing. Is that this nitrate? No, nitrogen. Oh, that's dissolved oxygen.
So, Dr. Chen, what exactly is the point of the nitrate test?
So basically, nitrate is a nutrient that is usually from fertilizers and manure storage, and when they run off into the water, they can provide nutrients for algae blooms. And the point with us doing the nitrate test is to make sure that there aren't too many nitrates within the water.
Tablet.
I
think
2
that means like
minutes. Yeah, 2. No. It just says hold up and down. So timer.
you have this on film, don't you?
And wait 5 minutes for the red color to develop. And then we compare the thingy to this. Note, wide range are sensitive to UV light. Protective sleeve will protect the reaction from UV light.
There's still
Indoors, there's no need to use protective sleeves. Okay. Next we can do... I don't know how you write the thing. Yeah, okay. Now we do the pH procedure.
It's in the river manual sheet.
Okay.
So in your guys, oh, I just told you.
The house.
8 ppm. You have to calculate percent.
Can I stop inverting now?
No, I put a timer on for 2 minutes. You have 37 seconds.
Can I stop now?
4, 3, 2, 1. Yes, stop. Okay. Now it says wait 5 minutes for the red color to develop. The pH is a measurement of the solute sales range. Fill the test tube to the 10 millimeter line.
Big reveal, Roger.
Going back to the thingy.
For science.
Wait, what was this? That was the nitrate one.
The red color did not develop.
Oh, what happens if it did not develop?
What's that for?
The nitrate procedure?
Oh look,
It's
the
stuck.
tablet did not.
There you go. How do you
Dissolve.
like dissolve? Yeah, it didn't dissolve.
No.
Just had to put it in the foil.
Rock back in the
Alright.
water. Be careful, because it's coming out.
What the hell?
Let's turn them.
So this is the red color that developed.
So that's what you want to.
That's 5. This is the red one here.
What are you doing?
This is nitrate.
Why'd you take it out of the
Because it's done!
Becca, why have I been doing this for 5 minutes?
It's like just now
Because
like
you have to
settling
dissolve.
down
You have
to
to
where
let it
like all the
dissolve.
stuff's going to the bottom, so I think we just gotta keep it.
There's nature on my paper!
I'm aggressively inverting it.
Okay, hold on. Temperature. Wear protective gloves at each site. Place the thermometer 4 inches below the water surface for 1 minute. And then, right after you're done with this, tell me what it is, and then go down there and do the same thing.
For science. It looked better from over there, over here.
How are you gonna get down?
Shut up, Roger.
So is that just
Would it be 1 or 2?
looks like a 2?
You think 2?
It's like in between, like 1.5.
Okay. Or 40 or 0?
It's 40.
You think it's 40?
Yeah, it's 40.
Okay.
Let's go science.
E. coli.
The lake outlet should have water that is the best compared to all of the other locations due to the water having its nutrients settled in the lake, the ditch, and the wetlands.
Are you recording?
Yeah.
Okay, so we're inside today for science because it's raining. No, it's not raining. It was raining. So we're indoors today for science. We have met. And I just filled that thing up. The DOD project. So we just filled that up, and the buckets over there are for the lake outlet.
The temperature is 14.2.
That's a lot warmer than it was yesterday.
Yes, it is.
Why? Oh, probably because it's indoors and has been warming up.
Wow, Charlie.
So smart.
Okay, and I see right there.
picture of E. coli? No, we're doing E. coli.
rock. Oh, there's a snail. Look, it's a snail. We got a snail.
Who is doing dissolved oxygen? Somebody volunteer.
That's dope. Okay.
10, dump it out a little bit. Is it at 10?
Definitely right.
Cool. Okay.
Out here trying to
Okay.
catch some feels.
You're so gross. Stop doing that. Please stop doing that. You did not just do that. Please tell me. Please stop doing that.
To figure out what's happening as a whole in that location, you have to have all the cue values.
What color?
Yellow.
You say 8.
Yeah.
Do you think 8?
Yeah, that's. I'm hoping that there's a. Put it in the center.
That's it.
I'd say 8.
I would go with an 8. Yeah. I don't think we have any.
Bless you. Yes, it is. Okay, here, I'll get the water. You get the other water sample.
Wait, Gavin, do you know anything
In fact, will you just stick your whole hand in there? Disgusting.
You're disgusting.
I use
Is it those?
the 10.
No, those are regular records.
So we're inside this little box, water in the outlet for us to test today. So we're testing pH, nitrate, phosphate,
Can you open?
and
What?
the dissolved level of oxygen. We're also going to be testing.
Open the door and he didn't even listen to you.
You just kind of looked at me like I was dumb.
I wonder why.
What's the result?
We can't decide if
Okay.
So what exactly is the point, and what exactly is the reason why we conduct the turbidity test, and what does it tell us?
So turbidity is when suspended particles within the water causes the water to be cloudy or murky, and that has an effect on photosynthesis in water plants because they need the light going through the water. And the test turbidity is just to see how cloudy the water is.
Can we get your opinion? Would you say this is 40 or 0?
Is to pick up the snail and doesn't know how to read because Roger doesn't know how to film and figure out the film. So on the sheet, if the tip is pointing up, the opening is on the right, then it's a right-handed snail. If the tip is pointing up, the opening is on the left, then it's a left-handed snail. So if you look at the snail, the tip is pointing that way, and the hole is on the right. So that means that it's a right-handed
moved.
snail.
If you want to know how to do that, talk to Aaron because he's the only one of you who knows. Don't shake it like that, Aaron. You're going to get a lot of bubbles.
You're just barfed. But it's kind of like a little blue. I don't know.
So that's 1.
That's what I was thinking.
That's 1.
That's a 1, right? Yeah, that's what I thought. Should I say it? The nitrate level is 5.
So what exactly is the point of the E. coli testing, and what does it tell us?
So E. coli is a species of bacteria, and most of them are dangerous. So, and E. coli comes from basically human manure. That's how we measure how polluted the water is, by looking for the level of E. coli. And when there is a high level of E. coli in the water, it's dangerous to people, especially people who are swimming in it, because the bacteria can enter the mouth, nose, cuts in the skin, ears, or eyes.
Fascinating.
So Charlie, why is the lake control worse than the lake inlet?
Well, Becca, this could be a result of a lot of different factors. If you look at our campus, it is very, very full of fields that are mowed very often. They maintain the grass, facilities do this, and we are right on the lake, which results in, whenever it rains, all of the grass clippings to flow right into the lake. This can cause for algae blooms and any increase in the algae in the lake. And another factor, which is right by where the lake control was, which may result in it being not as reliable, is the fact that there's construction happening just right out there, right where we did our lake control experiments. This could have runoff of the gasoline, different construction construction materials that they're doing flowing right into the lake, right where we did our testing, which could have a severe effect and severe impact on the results of which we got in our lake control versus the lake inlet.
Good answer. Why is the wetland outlet worse than the wetland inlet when it should be better?
This just proves and just goes to prove that the wetland is not working. It just simply is not doing what it is meant to do. This could be a result of many factors, too many to go into severe detail in, but it's just simply is not doing what it is there to do.
Why is the lake inlet so much higher than the wetlands outlet?
This is mostly due to the sediment that is flowing through the water and the material that is in the water flowing out of the outlet, and the large path that it travels through different streams, through different tunnels, on the bridges, through woodcraft. It all settles at the bottom. It has a very long time to settle and just get out of the water. So majority of the nutrients, majority of the sediment settles down into the bottom of the river.
Thank you. Why is the wetland outlet worse than the wetlands inlet when it should be worse? Wait, that was not the right question.
No, that was not the right question.
Are you ready? Yeah. Okay. Why is the wetland outlet worse than the wetlands inlet when it should be better?
So the wetlands outlet is much worse than the wetlands inlet simply due to the amount of rain and the substantial just overflow that's happening within the wetland itself. It's not allowing it to settle, it's just being overwhelmed with all of the rain we've got. So. Thanks for staying quiet, that was great.
Yeah, seriously, thanks.
So, Raj Daddy, what exactly, I am here today with Roger Chen, the prestigious doctor in biology, and we are here to talk about today the effects of eutrophication and some certain tests and why exactly the tests are conducted. So would you be able to tell us a little bit about that and introduce yourself? We are here today, by the way, in the prestigious Maine Barracks Science Testing Facilities. So our first question for you today, Roger Chen, is, Dr. Chen, is what is the point of testing?